Good morning, everyone. I'm Dr. S.K. Singh, and I'm pleased to welcome you today. Today, we'll be exploring gaseous sterilization, a fascinating and important topic in the field of sterilization techniques. On this first slide, you see my name and credentials, and the main focus of our presentation. Over the next few slides, we'll cover the principles, methods, advantages, and challenges of gaseous sterilization. So, let's get started and dive into this essential subject. Welcome to the second slide. Today, we're going to talk about gaseous sterilization. This method involves using chemical gases or vapors to eliminate microorganisms. It's especially useful for sterilizing devices that are sensitive to heat and moisture, which can be damaged by traditional sterilization methods. Some of the most common sterilants used in this process include ethylene oxide, or EtO, vaporized hydrogen peroxide, often written as VH2O2, and hydrogen peroxide plasma. Each of these has unique properties and applications, but they all serve the same purpose: effectively sterilizing sensitive equipment. When we look at the different types of gaseous sterilization, ethylene oxide is perhaps the most widely used. That's because it has a high penetration capability, meaning it can reach into small or intricate devices to ensure thorough sterilization. Vaporized hydrogen peroxide and hydrogen peroxide gas plasma are also important, offering their own advantages, such as shorter cycle times and lower residues. Overall, gaseous sterilization plays a crucial role in maintaining safety and sterility, especially for devices that can't withstand traditional heat or moisture-based methods. It's a key part of our sterilization toolkit, ensuring we can sterilize a wide range of medical and laboratory equipment effectively. Let's take a closer look at the key process parameters that influence sterilization effectiveness. Temperature, for example, especially in ethylene oxide or EtO sterilization, typically ranges from about 30 to 60 degrees Celsius. This range is crucial because it helps optimize the sterilization process without damaging the materials. Gas concentration is another important factor. Getting the right amount of EtO ensures effective sterilization while maintaining safety. Relative humidity also plays a role, as it can influence the reaction rate. Exposure time is equally vital. Longer exposure can improve sterilization, but must be balanced to prevent material degradation. Pressure or vacuum cycles are used to help the gas penetrate complex devices and materials thoroughly. Now, what's key here is that sterilization isn't just about one parameter. It's the combined effect of all these factors working together that determines whether sterilization is successful. Moving on to how these processes work at a molecular level, the mechanism of action differs depending on the sterilizing agent. For EtO, it alkylates DNA and proteins, which effectively prevents microorganisms from replicating. In the case of hydrogen peroxide or plasma sterilization, the process involves free radical oxidation, which destroys cellular components. These mechanisms lead to critical outcomes: protein denaturation, damage to cell membranes, and ultimately, cell death. So, in essence, whether through chemical modification or oxidative damage, these processes ensure that harmful microorganisms are effectively eliminated, aligning with our overall goal of achieving reliable sterilization. Now, let's take a closer look at the ISO 11135 requirements for ethylene oxide sterilization. This standard covers several key areas, including process development, validation, and routine control. These steps are essential to ensure the sterilization process is both effective and consistent. When we talk about validation, it's broken down into 3 life cycle stages: IQ, OQ, and PQ. IQ, or installation qualification, confirms that all equipment is installed correctly. OQ, or operational qualification, verifies that the equipment operates within specified parameters. And PQ, or performance qualification, demonstrates that the process consistently produces the desired sterilization results. The ultimate goal here is to ensure a reproducible sterilization process, one that works every time, reliably. Now, moving on to the regulatory framework. There are several global standards that support sterilization practices. ISO 11135 is specific to ethylene oxide sterilization. ISO 14937 covers general sterilization processes applicable to various methods. ISO 22441 addresses sterilization using vaporized hydrogen peroxide, and ISO 13485 relates to quality management systems for medical devices. These standards are supported by regulatory bodies around the world, such as the FDA in the United States, the EU Medical Device Regulation in Europe, and CDSCO in India. It's important to remember that compliance with these standards and regulations is mandatory if you want your medical devices to be approved and marketed in these regions. Keeping up with these requirements ensures not only regulatory approval, but also the safety and efficacy of your sterilized products. Now, let's take a closer look at the ISO requirements for H2O2 sterilization. The key standards here are ISO 22441 and ISO 14937. These standards provide a comprehensive framework that covers several critical aspects of the sterilization process. First, they define the process itself, ensuring that everyone is on the same page about how sterilization should be carried out. They also specify equipment qualification, which means verifying that the sterilizers are capable of achieving the desired sterilization conditions consistently. Performance validation is another crucial element, confirming that the entire process actually works effectively over time. Monitoring plays a vital role here, and these standards require continuous oversight of temperature, pressure, and cycle mapping. This helps ensure that all conditions stay within the validated parameters, maintaining process integrity. Moving on, let's talk about sterility assurance level, or SAL. This is essentially the probability that a sterilized unit is non-sterile. The standard requirement for SAL is 10 to the power of -6, or 10. In other words, there's only a 1 in a million chance that a sterilized item isn't sterile. To demonstrate that this level of sterility is achieved, we rely on biological indicators and validation studies. These tools provide the evidence needed to confirm that the sterilization process consistently meets the required standards, giving us confidence in the safety and reliability of the sterilized products. All of these elements come together to support our overall goal, ensuring patient safety through effective, validated sterilization processes. Now, let's take a closer look at the validation approaches we use to ensure our sterilization processes are effective. We typically rely on two main methods, the overkill method and the bioburden-based method. The overkill method is quite conservative. It involves applying a sterilization cycle that's designed to kill even the most resistant microorganisms. This approach provides a safety margin, making sure that if we follow the process correctly, sterility is guaranteed. It's often used when absolute certainty is required, especially in high-risk situations. On the other hand, the bioburden-based method tailors the validation to the actual microbial load on the items before sterilization. Here, we assess the initial microbial contamination, what we call the bioburden, and then validate that our sterilization cycle can handle that level effectively. This approach can sometimes be more efficient because it's based on real-world conditions rather than worst-case scenarios. Now, within these validation strategies, a few key elements are crucial. First, biological indicators, or BIs, play a vital role. These are standardized tests involving microorganisms that are highly resistant to sterilization processes. We use BIs to verify that the sterilant has penetrated and effectively killed microorganisms. Next, we perform half-cycle studies, which involve testing at different points during the sterilization cycle to ensure each phase is working properly. This helps us pinpoint where validation is effective and where adjustments might be needed. Finally, worst-case load validation is about challenging the sterilization process with the most difficult loads to sterilize, those with the highest bioburden, the most complex configurations, or the most resistant microorganisms. Validating under these conditions gives us confidence that the process will work reliably under normal circumstances. All these elements tie back to the overall goal, ensuring patient safety and product efficacy through rigorous, scientifically sound validation methods. Now, let's take a closer look at hydrogen peroxide plasma sterilization. The process begins with injecting H2O2 vapor into the chamber. Once in the plasma phase, the vapor generates free radicals, which are highly reactive species that effectively destroy bacteria, viruses, and spores. An important point here is that the byproducts of this process are simply water and oxygen, which means there are no toxic residues left behind. This allows for faster cycle times, typically under 1 hour, making it a very efficient sterilization method. To give you some context, this process is similar to other sterilization cycles like ethylene oxide or ETO, but with some key advantages. The cycle includes steps such as preconditioning, which involves humidity control, vacuum cycles to prepare the chamber, gas injection, the actual exposure phase where sterilization occurs, and finally, aeration. The aeration step is critical because it removes any residual hydrogen peroxide to meet safety standards, specifically those outlined in ISO 10993. This ensures that the sterilized items are safe for handling and use. Overall, hydrogen peroxide plasma offers a combination of effectiveness, speed, and safety, aligning well with modern sterilization requirements. Let's walk through the calculation of exposure time for SAL. First, we start with our bioburden, which is 10 to the power of 4, or 10,000 organisms. The required SAL, or sterility assurance level, is 10 to the minus 6, meaning we need to reduce the microbial load to 1 in a million. To achieve this, we need a total log reduction of 10 logs. Specifically, 4 logs to go from 10,000 down to just 1 organism, and then an additional 6 logs to ensure safety, reaching that SAL target. If the D-value, which is the time needed to reduce the microbial population by 90%, is 2 minutes, then the total exposure time required is simply 10 logs times 2 minutes per log, giving us 20 minutes. Now let's clarify the D-value a bit more. The D-value equals the time it takes to achieve a 90% reduction in the microbial population. For example, if we start with 1 million organisms, 10 to the 6, and after 2 minutes, the count drops to 100,000, 10 to the 5, that confirms that our D-value is indeed 2 minutes. So, understanding this D-value helps us plan the exact exposure time needed to reach our sterilization goal efficiently. Let's talk about some of the key risk and safety considerations when working with these sterilization methods. First, for ethylene oxide, or ETO, it's important to remember that it's toxic and carcinogenic. This means we need to handle it with extreme care to protect our health and safety. Because of its hazards, there are strict emission limits in place to control its release into the environment and minimize exposure. Now on to hydrogen peroxide, or H2O2. It's considered a safer alternative compared to ETO, which is a big advantage. However, it does have limitations, such as limited penetration, meaning it might not be suitable for sterilizing very complex or dense items. Lastly, regulatory requirements play a crucial role across the board. We must prioritize worker safety by following proper protocols and using protective equipment. Residual limits are also in place to ensure that any leftover chemicals on sterilized items are within safe levels. And of course, environmental compliance is essential to prevent any harmful impact on our surroundings. Connecting this all back to our overall theme, understanding these safety considerations helps us choose the right sterilization method responsibly, balancing effectiveness with safety for people and the environment. Let's take a closer look at the advantages and limitations of these sterilization methods. First, on the advantages side, both ETO and H2O2 are particularly suitable for delicate devices. That's because they operate at relatively low temperatures, which helps prevent damage to sensitive equipment. This makes them ideal choices when high heat could compromise the device's integrity. Now, turning to the limitations, there are some important considerations. Ethylene oxide, or ETO, requires a long aeration time after sterilization. This is necessary to remove residual gas and ensure safety before the devices can be used again. It can be a bit time-consuming, which might impact workflow efficiency. As for hydrogen peroxide, or H2O2, one challenge is its limited penetration. It might not reach all areas inside complex or densely packed devices, which can affect sterilization effectiveness. Plus, both methods require complex validation processes to confirm that sterilization is complete and consistent, which can add to the overall complexity and cost. Understanding these advantages and limitations helps us make informed decisions about which sterilization method best fits our specific needs, especially when balancing device integrity, safety, and efficiency within our overall sterilization strategy. So, to wrap up, gaseous sterilization plays a crucial role in ensuring the safety and effectiveness of modern medical devices. It's a process that's heavily regulated by international standards like ISO, as well as various global health agencies, to make sure everything meets strict safety criteria. Validation is key here. It confirms that the sterilization process consistently achieves a sterility assurance level of 10 to the -6, meaning there's only a 1 in a million chance of a live microorganism surviving. And an exciting trend we're seeing is the shift toward lower toxicity methods, like hydrogen peroxide plasma. These newer methods aim to reduce potential risks to both patients and the environment while still maintaining high sterilization standards. All of these points highlight the importance of continuous innovation and regulation in sterilization to keep pace with evolving medical technologies and safety requirements. For further support, please don't hesitate to reach out. If you have any questions or need additional information, we're here to help. You can contact us through the provided channels. Thank you so much for your time and attention today. We truly appreciate your interest and engagement. Remember, we're committed to supporting you every step of the way. Again, thank you and have a great day.